Let me get my speaker. So, thank you very much. It's really an honor to be here today, and I'm going to be talking about bringing precision medicine to smoking cessation. And I think that John Newport Langley would be right here with us today, going forward and wondering what his genotypes were. So I'm going to really go over many different aspects, and as I was organizing my talk, I was thinking of it as different chapters in the story of how we're getting to precision medicine. And so let me start with chapter 1, which is the brave new genetic world. The brave new genetic world is really this paradigm change which has occurred in the past 15 years. So what I often think about now, and as I'm teaching students, is I think of 2005 as time zero. That was when this new era in genetic studies came about because of the sequencing of the first human genome. It was really a tremendous amount of work, cost over $1 billion, and at that time, really one of the first editorials came out asking, was the human genome effort really, was it really worth it? We've just spent so much money on this science, and, what are we going to get of it? So very quickly, this was answered. Later that year, the first genome-wide association study, which reached this very high level of significance, our significance levels that we've set in these studies as 5 times 10 to the minus 8th p-values, it identified strong genetic contributions to age-related macular degeneration. This is a common disease, affects the elderly, and leads to blindness. So very quickly, this new genes with variation in it, which leads to different risks associated with this disease, were found. So to demonstrate the different types of findings that we've had, the NHGRI, the National Institute of Health Human Genome Research Institute, has been putting out this catalog. So this is the catalog in 2005. What you see here are the human chromosomes, and this little circle represents that finding for age-related macular degeneration. In 2006, we had added 3 more variants, so there's a total of 4 genome-wide associations findings across all of human diseases. 2007, you see some more. 2008, we're on a roll. 2009, it keeps on going. 2010, even more. 2011, 2012, 2013, 2014, 2015, 16, and here we are, 2017. So really, we have witnessed in this past decade a revolution in genetic technology. We've seen genotyping go down to really pennies a genotype. We could genotype individuals for less than $100. We have identified genetic variants that contribute to longevity, height, weight, obesity, diabetes, hypertension, the list goes on. Every type of characteristic that you could think about in humans now has genetic variation associated with this. And this genetic variation is just wonderful. It makes us all different. It makes, just who we are, the diversity in the world. So we really need to be embracing this type of genetic contributions to all of our characteristics. So with this, we don't want to leave the field of addiction and smoking behind. And so as these studies were coming about with the Human Genome Project, we were starting to work on our study design. And this study design was as part of the collaborative genetic study of nicotine dependence. So classic studies, some by Gary Swan, really were demonstrating that genes do contribute to different types of smoking behaviors. They contribute to heaviness of smoking, smoking initiation, a variety of constructs with smoking. And so in an airport conference room, Dorothy Hatsukami, Eric Johnson, Ted Reich, Naomi Breslau, several others, we sat around trying to design this study. This was a multi-site study across Detroit, Minnesota, and St. Louis, and we argued for a full day about what our model of a nicotine use disorder would be. And we kind of came upon this cascade of behaviors that you start from never use. There's this initiation where you have your first puff. You then transition to become a smoker, defined by smoking 100 cigarettes in your lifetime. And then some individuals transition to nicotine use disorder. We made a strategic decision at that time that we were going to focus on this transition from smoker to nicotine use disorder as the single area that we would study in this project. So trying to understand this flip. And in part, this was led by many of these twin studies, which were showing that genes and environment contribute to all of these steps. But environment has a much stronger role in these initiation phases. Peers are important, parental smoking is important, taxation is important. All of these things are very important with the initiation phase. But the strongest genetic effect was in this transition from being a smoker to going to nicotine dependence. That's when biology took its strongest hold. So what we did then is we set about calling people on their landlines. So this is in the early 2000s, and we screened over 50,000 people in the St. Louis and Detroit area. What we found was that about half of the population had smoked a cigarette, had a puff. It was almost 60% transitioned from this puff to becoming this definition of a smoker by smoking 100 cigarettes in their lifetime. And then three groups really came out of the smokers. There were those individuals who had no symptoms of dependence. These are often the chippers that you talk about, the individuals who never smoked more than 10 cigarettes a day. They could give it up at any time. They tended to smoke later in the day. And these individuals often aren't coming into our clinics with their healthcare problems. There's another group that had some symptoms of nicotine dependence. And then there was this group with clear nicotine use disorder. They had high scores on their Fagostrom test. And so we selected individuals for genetic study from these two extremes, those with current nicotine use disorder and those who never had any symptoms of nicotine use disorder in their lifetime, even though they were smokers. So, what did we find? We found that variation in the nicotinic acetylcholine receptors, that variation is the strongest influence on how one smokes cigarettes. And I use that very general words of how one smokes because it really contributes to a lot of different behaviors with smoking. So we did this, at that time, large-scale genetic study. We had 3,713 SNPs tested, which now is just kind of humorous that it's easy to get 1 million done with much less cost. And our study was quickly followed by other very important studies in the field that confirmed the finding of the chromosome 15 region associated with smoking. Wade Berrettini identified this, and then Thorgeir Thorgesson from Decode also identified the same region. This was then followed by further large-scale collaborations around the world. And so let me show you kind of what the findings look like. So this is what we generally call a Manhattan plot. Because you're supposed to see the skyscrapers of Manhattan. I always call this one the Dubai plot, where we see something really sticking out of the plot. So what we have here on the y-axis is negative log of the p-value. So the higher it goes, the greater the significance value. So we have a significance value of p 10 to the minus 35th with the chromosome 15 region. You see, nothing else even reaches genome-wide significance in this study of over 80,000 individuals. And this chromosome 15 region is just sitting smack dab on top of a cluster of nicotinic receptors. And so God looked down on us and smiled and really kind of made it easy for us of this is where you should look. So now let's go into a little basic science here. So what do we know about this receptor group? So there are actually 3 nicotinic receptors, subunits sitting in this region, the alpha-5, the alpha-3, and the beta-4 nicotinic receptors. They're all in a row. This is how our genome is often arranged, with clusters of genes and often clusters of similar genes. We know that the nicotinic receptor has its protein structure. We know that the receptor subunits form pentamers, which then conduct ions through the membrane. And what we see is this variant, which we often called Mr. Big. This was in the time of Sex and the City. So Mr. Big is this variant in the protein coding region of alpha-5. And this change causes, this change in DNA variation causes a change in the amino acid of the alpha-5 nicotinic receptor from aspartic acid to asparagine. So again, we look back at this and it's so common nowadays, but we were just at the beginning of looking at cross-species conservation. So as we were sequencing the human genome, sequencing was coming out for a variety of different animals. So we have here Homo sapiens at the top, but it goes down to cow, rat, mouse, chicken, a variety of things. And what we see is this aspartic amino acid is conserved across all of these species. But it's in humans that we have this change, that some people have this aspartic amino acid in their protein, some people have the asparagine. So but when you see this type of conservation across species, again, it's generally telling us something. And what it's telling us is that this is really important. With evolution, we're not going to allow change, and we're going to make this protein structure and in this region look the same as it is in a rat, the same as it is in a cow, the same as it is in other primates, a variety of different animals. So again, this type of evidence was kind of leading us to think, again, this change is probably a causal variant. Through work with Jerry Stitzel at the University of Colorado, he then induced this change into an in vitro system and showed that this single amino acid change alters the response of nicotine. It alters the response of the nicotinic receptors and its conductance. We also were then able to go and look at the expression of this. So through Jen Wang and Alison Goet's lab, we saw that not only is there an amino acid change, but we could also see a change in expression of the protein. So there's not only this amino acid change, but multiple other changes in this genetic region are contributing to nicotine dependence. So work through that was led by Emily Olson. Then we moved on with sequencing, and what she found was that not only are there common variants in this region, but there are rare variants that are changing this protein, and rare variants that are unique to some individuals. So in this schematic, all of the purple variants here were not seen in any of the other databases of human genomes, which at that time were over 100,000. So again, we have a tremendous amount of diversity, and this diversity is leading to differential risk of smoking. So this whole series of studies really kind of nailed it, that this chromosome 15 region, which encompasses 3 nicotinic acetylcholine receptor subunits, the alpha-5, the alpha-3, the beta-4, are associated with nicotine use disorder. They're associated with heaviness of smoking, so how many cigarettes you smoke per day. They're also associated with the smoking intensity. Other studies that we've done have controlled for how many cigarettes you smoke per day, and what we could see is that the amount of CO that a person has in their on exhalation is associated with this variant. So this variant is changing multiple different behaviors. We know that there's this common variant. It changes the protein structure. It's present in about 50% of the population. We know that there's a second common variant, again, at about 50% of the population, that changes expression of this gene. Then there are many less common variants that change this nicotinic receptor and also contribute. So these are go from rare to common variants, altering how people smoke, how many cigarettes they smoke per day, their intensity of smoking. And so with this, we have to ask the next question, what happens with disease? So studies were then undergoing at the same time genetics of lung cancer and chronic obstructive pulmonary disease. So one thing that I love about taxation is that we could track per capita consumption of cigarettes in the United States. And cigarette smoking is really a 20th century behavior. It started at the beginning of the 20th century. We think of the mass production of cigarettes. Consumption started increasing and then peaked in 1964 with the first Surgeon General report. We stayed at about that same level of per capita cigarette consumption for about 20 years until we finally had further regulation, Clean Indoor Air Acts, additional taxation, which has now dropped per capita cigarette consumption dramatically in the United States. I could also look at lung cancer death rates in the US, and this graph is really amazing at how it mirrors per capita consumption. So lung cancer, again, a very rare disorder at the beginning of the century, and didn't really start becoming more prevalent until the 1930s. And that really reflects the initiation of smoking that occurred during World War I, and the soldiers initiating smoking during their time in the service. We have this lag of 20 to 30 years, but again, the decrease is coming, and lung cancer nonetheless remains the greatest cancer death in the United States. So genetic studies have been looking at the contributions to lung cancer. We know that lung cancer runs in families. We've known that there's this genetic variation. And in 2008, shortly after our first studies on smoking came out, the series of studies on lung cancer came out, and it was extremely controversial. The reason it was so controversial is because of this finding. So if you recall the Dubai plot that I showed you for smoking, this is the Dubai plot for lung cancer. And what you see is the greatest genetic contributor to lung cancer is the genetic contributor to smoking. So the controversy behind this was that we kind of knew that smoking caused lung cancer. And the lung cancer people were not looking for the genetic contribution for smoking. They were looking for the genetic contribution of cancer. So an argument went about for about 5 years. I think that it's pretty much settled now, is that this finding for lung cancer really is the finding for the genetic contribution for smoking. So that strongest genetic contributor is related to smoking behavior. Nancy Ciccone collaborated with the lung cancer group and also the chronic obstructive pulmonary disease group, because they had basically the same type of graph. And we showed that these genetic variants were identical, the same genetic contributions to lung cancer and chronic obstructive pulmonary disease are the genetic contributions to smoking behavior. So we have this kind of interesting wraparound of smoking, lung cancer, genes, all coming down to the same chromosome 15 region. But as I think about this, I really think, what is the ultimate goal? The ultimate goal really is smoking cessation. How could we reduce smoking amongst the population? We know that over 40 million Americans continue to smoke. We know that smoking is increasing worldwide, and over 400 deaths occur every year in the United States related to smoking behavior. So let me go back to our model that we had here with smoking cessation. We think of becoming a smoker, going on to nicotine dependence, but we have two different paths going to smoking cessation. There's those who are nicotine dependent, who are quitting, and those who are the kind of lighter smoker, not dependent, quitting. In general, the group that we tend to be working with are those who have the greatest difficulty with smoking cessation, and that's that group with nicotine use disorder. So I've been lucky to work with Tim Baker, Michael Fiore, Megan Piper, and that team with the University of Wisconsin Center for Tobacco Research, and it's been really a pleasure kind of layering on top of their expertise with smoking cessation some of our genetic work. So Lishan Chen then led this first paper, which looked at genetic variants that predict smoking cessation risk. And what she found was there was this interplay of these genetic variants. Those at the highest genetic risk for heaviness of smoking, lung cancer, COPD, were those who then had the greatest success with pharmacologic treatment for smoking cessation. And in placebo groups, this group had by far the worst outcomes. So those at highest risk for heaviness of smoking, highest intensity of smoking, also really couldn't quit in this trial. But the data are not quite as clear yet for smoking cessation. And I have here the data back to back. The first one on the left is data from the trial from the University of Wisconsin, looking at nicotine replacement therapy. The trial on the right is actually the Pfizer trial for varenicline. We were able to obtain their genetic data that they had on their trial and analyze those data. So we see this kind of interesting difference. One, and I would say the major issue with these two different trials is the placebo group. We see from the University of Wisconsin, their placebo group has this clear kind of decrease. The placebo group is the light gray bars on kind of each of the three genotypes. So as you move from low genetic risk to higher genetic risk, your likelihood of abstinence greatly decreases. And if you are given nicotine replacement therapy, your likelihood of abstinence with treatment, which are the speckled darker bars, increases. In the Pfizer study, kind of didn't matter what your genotype was with the placebo group. Their overall placebo group rate was about 20% end of treatment. And then with treatment, we see this, again, this trend with the high genetic risk. So what I really think is going on here is environment matters, that the intensity of treatment really allows the expression of genetic variation. There's a whole other series of papers looking at pregnancy. So women during pregnancy, those at high genetic risk, are the least likely to quit. But as you add lots of environmental aspects to it that are not intense, environment kind of takes over and wins over biology. Nonetheless, I think we can still see this delay in smoking cessation at the population level. So with our colleagues in the lung cancer field and general population field who have large genetic samples, we were able to look at the age of smoking cessation. And what we found with this amino acid change, that if you have the high-risk genetic variant, your mean age of smoking cessation is 4 years later than those at the lowest genetic risk. We also found that your median age of the development of lung cancer, so if you have lung cancer, is 4 years earlier. So both of these findings have lots of clinical impact. So 4 more years of smoking, heavier smoking, greater intensity is driving this risk of disease. And when we look at this median age of lung cancer diagnosis, we know that over 50% of people who are diagnosed with lung cancer die within a year. So this difference in the median age also is making a large clinical difference. So now I get to move to kind of the future. So I've summarized a whole series of studies done over the years, but there's more. There's much more than that. The chromosome 15 region with the alpha 5, alpha 3, beta 4 has multiple variants in that region. The large-scale genetic studies now include over 1 million people. These studies are being led in this GSCAN consortium by Scott Vries, and we know that there's at least 10 variants in this region that individually contribute to the risk. We also know that variation in other nicotinic receptors are also increasing the risk of heaviness of smoking. And then the other elephant in the room is nicotine metabolism. So the chromosome 15 region with the nicotinic receptors, the chromosome 19 region of nicotine metabolism, those are the two main drivers of heaviness of smoking, smoking cessation, smoking intensity, lung cancer, COPD. And so we actually have, as a result, an incredible amount of knowledge and clear targets for us to move forward with. So, precision medicine. What does this mean? Precision medicine is the idea that we identify individuals, we give them the treatment that is best for them. We think of genetic variation with it, but we should also think of environment. Environment is incredibly strong. And what's wonderful, I think, about the genetic findings are that it helps us identify the environments and hone in on specific environments that are good and bad for individuals. So we're starting to understand that interplay between the genes and environment. So we're challenged now of how do we move all of this knowledge that we have. We're at hundreds of genetic variants that contribute to different smoking behaviors. How do we translate this into the community? So there are really, we've been thinking about this quite a bit over the last several years, and now I get to speculate about what I believe the future will bring. When we move tests out from our science world into kind of care and the public, we think of three major aspects. We think of analytic validity, clinical validity, and clinical utility. Analytic validity is when you do a test, how accurate is the test? Can you do that test again and again? And that is absolutely nailed at this point. There is no doubt with the genetic technologies that we have, we could analyze people, identify it clearly, consistently, with incredible precision. So check, nailed. Clinical validity. Clinical validity is the idea of when you do this test, what does it tell you? Does it give you something that is meaningful? I think we have this down also. We know that these genetic variants increase your risk of lung cancer. If you have, again, with the simple story of that one amino acid change, the Mr. Big, we know that that increases your risk of lung cancer by 30%, even after controlling for your smoking behaviors. We know that these genetic variants contribute to your risk of COPD. We know that these genetic variants contribute to a delay in smoking cessation. We know that these genetic variants contribute to the heaviness of smoking and smoking intensity that you have. This is very, very clear. So, check on clinical validity. The third aspect that we think about is clinical utility. And clinical utility is that idea of, can you use this to change? Can you use it to change your prescriptions? Can you use it to change smoking cessation? Can you use it in a way to change behavior? This is where we do not have the evidence yet. But I think that nonetheless, the knowledge of risk is enough for us to move it forward. So, precision medicine for smoking cessation, I think, has three main characteristics. It has this idea of risk stratification. We can tell individuals now what their risk is. And we are sitting on this knowledge right now, and it is over and above how much they smoke. So should we be telling people this information? I think knowledge wants to be free, and yes, we should start embracing this idea of giving this idea of risk stratification forward. Does it change our treatment for smoking cessation? At this point, what I know is that about 70% of Americans who are smoking want to quit every year. I know over 50% try every year, but only 30% use evidence-based counseling or pharmacologic treatment. So really we have this incredible under-treatment at this point. Whether we're going to be able to choose specific treatments based on this genetic risk, I don't know. I think there's some hints to that. Whether it will motivate individuals to change their behaviors, to move forward and use more effective medications. That is an unknown, but this is where we should be moving. So I think we're living in disruptive times here. We're living in disruptive times. There's been a lot of discussion about electronic cigarettes, and I'm going to talk about genetic services that are direct to consumer. So 23andMe is out there. It's a private company. I use them in my studies, but I have no affiliation. I buy their product, but this product is amazing genetically. For $99, you spit in a tube, and you could get your ancestry, and you could get your health risks. The FDA kind of had a little thing with them a few years ago, but they are up and running and quite effective at this point. So their genetic service is outstanding. Here's my ancestry. I'm of European ancestry, kind of knew that already. But what we could also see is we could see our different types of disease risk that we have. 23andMe is now returning to direct to consumer, their genetic health risks, ancestry, wellness, carrier status for some Mendelian type disorders. And what I anticipate will happen in the future is I think that smoking is going to come under one of these wellness reports. That it's the idea of, kind of the behavior, how you're living, whether you are drinking alcohol, consuming caffeine, weight, your lactose intolerance. So this information wants to get out there, and a company is going to move it out there. And I'm not afraid of that. I think that that will be a good thing. So what do people want? What I know is over 1 million people have purchased 23andMe. These reports are out there. As we've done focus groups with individuals, they want to know what their genetic risk is. As we've piloted returning genetic information to people, everyone says, I love it because it's all about me. And so I think what we're going to see is we're going to see this movement there, in some ways similar to how we've seen the population move to electronic cigarettes. And I think we need to quickly catch up to them and understand what's happening as people are getting their genetic risk, and what does it mean, and how could we utilize this best. So I want to move now to my acknowledgments. So I got through the science of this, and I'm just so happy to be here today with the receiving this Langley Award. I found this picture, and I just want to highlight something in this picture, which is John Newport Langley. Looks like he was a smoker. I'm sure that, given the population prevalence of these things, I'm sure he had some of the high-risk genetic variants associated with smoking. So I think he would be sitting here today wanting to know what his genotype was, what his risk was. And I've worked with, this is really team science. This is part of the list of all the individuals that I've worked with. These are some of my friends. Gary Swan, Dorothy Hatsukami is here, Eric Johnson, Tim Baker, Sarah Hart, Li-Shun Chen. Ted Reich is no longer with us starting this. But I just want to say that I really have been blessed working with this group, because they've taught me so much over the years. And again, this work is team science, and let's go out there and make a difference in the world. So I want to thank you all very much, and I will be happy to take any type of questions.
That was fantastic. Thank you. We're really privileged to have you here today. In your language, and also when Dorothy introduced you, the language was association rather than causation of the genes. And I'm wondering if you could talk some about what you see and where the science is in terms of, is it that these genes start off and that leads to somebody being a greater risk for developing nicotine addiction, or is it that the assault of tobacco use potentially changes genes and then becomes a marker of heavy tobacco use?
Right. So this is a great question, and it's the question of association, causation, and changes. So all of the science that I was talking about today is actually the genes that we were born with, the genetic variation that we were born with. And we use the word association, and that has to do with the genetic architecture of us as humans, that there's multiple SNPs that are correlated. So we have this association, that is clear, but which exact variant is causing the change is some of the science that needs to go on further. You raised the question about does smoking change the way our DNA works? And the answer to that is clearly yes. So these are called the epigenetic changes that occur. And what we know now is that we could identify smokers who've quit 20 or 30 years ago with their DNA, that some of these changes are long-lasting throughout their lifetime. So these DNA signatures, these epigenetic changes, which is related to methylation of the DNA, many of them go away when you quit, but some are there basically forever. Yes, I think there's some questions over there.
This is on. Hillary Tyndall from Vanderbilt. Thank you so much for your talk. It was really provocative, and I appreciated the suggestion and encouragement to get out and do and try to apply some of this work. We have had limited experience at our institution with applying nicotine metabolite ratio, not genetic testing, but a simple blood test. We were very grateful to NTR for publishing this work last fall. Again, just a pilot, but it's exactly as you say, the patients love it, smokers love the idea that it's all about them. There was high feasibility, and only in about 15% of people was their fear expressed for knowing their metabolism, knowing if it would be harder for them to quit. But we'd like to move forward with this, and I'm just very grateful for your encouragement to go ahead and apply it clinically, because that's really been a question that's come up at SRNT the last few years. Do we study this more? Do we apply it? Which direction should we go? Thank you.
Thank you. I agree. Let's apply.
So Stan Glantz from UCSF. So I just wanted to, are you saying, excuse me, that the presence of this gene is making people more susceptible to developing heavy smoking, which then exposes them to a bunch of carcinogens, which then those carcinogens cause the cancer,
Yes.
the lung cancer? Or are you saying that that there's also, in addition to this gene being associated with heavy tobacco use, it's also somehow directly affecting cancer risk in the lung, in the cells in the lungs themselves, or both?
I am saying the first.
Okay.
Which is this gene, the variation in this gene leads to heaviness of smoking, leads to increased exposure of the toxins associated with smoking, leads to lung cancer. So there's still a little controversy in the field, but I would say, like climate change, there's always a little controversy, but the vast majority of the field believe that it's really related to the smoking behavior.
Okay, but it's not something happening in the lung tissue. Okay, thank you.
And the strongest evidence of this is that there are people with lung cancer who have never smoked, and this genetic variation has no effect. There's no association at all, and that's the strongest evidence we have.
Thank you.
Dr. Bierut, thank you for this outstanding presentation. I'm Dr. David Sachs, a pulmonary physician and director of the Palo Alto Center for Pulmonary Disease Prevention and a member of the recently formed working group of the American Thoracic Society to develop the first evidence-based clinical practice guideline for assessing and treating tobacco dependence in several decades. This should be out in about 2 years. We're just starting. My question is a follow-up to Dr. Glance's, and that is this. I think it was 1955 when the tobacco industry was doing everything it could to discredit the notion that cigarette use was in any way linked or associated with cancer. A scientific paper was published by a statistician by the name of Fisher, same man who I believe developed the Fisher exact test.
Right.
And he raised the hypothesis that there is a gene that causes smoking, and that same gene causes lung cancer. So I think what I hear you say is that now you have the evidence that the Fisher hypothesis is disproved, because what I hear you saying is there are families of genes on chromosome 15 that will, if you have the wrong genes, you will be at risk to develop tobacco use disorder. There are then other genes on chromosome 15 that if you are unlucky enough to have those, you will then be at much higher risk to develop lung cancer, and you're talking about the intersection of these two families of genes. Do I have that correct?
They're identical. They're not two different groups. They are identical.
So then the Fisher hypothesis is proved. He was correct. Is that right?
I think it's so. What I believe, there's something called pleiotropy in genetics, which is the idea that a gene could cause smoking and this gene could cause lung cancer. And we should think of this as these genetic variants lead to increased smoking intensity, deeper inhalation, lack of smoking cessation, and all of that increased exposure to the toxins leads to lung cancer. It is all through the smoking behavior. It is not a direct effect.
Thank you.
So I think it's the opposite. And Fisher was a smoker. I have a wonderful photo of him too. I think it is disproved.
Linda Ferry from Loma Linda University, and I have a comment on what you said. Should we use this for smokers? I've been tremendously benefited from my membership in SRNT and learning from people like you working in different fields from me. And I have been using this information about the genetics of smoking in the patients I have treated since 1985 at the VA hospital. In the VA hospital, we have lots of smokers, okay, by military experience and PTSD, et cetera. And I'll tell you that when I share with them the information that you and your colleagues have done on genetics, their eyes pop open and they say, really? It's not because I have some lack of willpower and character deficit. It's because I'm genetically addicted. It changes their whole attitude toward the approach of quitting. So I have treated over 15,000 smokers at the Loma Linda VA since 1985, and on a day-by-day basis, I use the information that I learn here at SRNT with my smokers, and it makes a huge difference. So my question is, in the early days, I did some research with Dr. David Cummings at the City of Hope in California on the DRD2 receptor. Could you make a comment on that? Because in our paper that we published on that, we found that same correlation of people smoking much heavier, starting much earlier in life, et cetera.
So what I would say is the modern genetics really is not focused as much as DRD2. DRD2 is a dopamine receptor that received a lot of examination. Some studies showed it, and most did not. In the modern genetics field, we think that most of those, the findings were false positives. So it's the nicotinic receptors, it's nicotine metabolism. Those are the two major drivers in smoking behavior.
Yes, hi, Adam Goldstein, University of North Carolina. Thank you. I want to ask a question. So we see about 4,000 tobacco users a year in our nicotine dependence program, the majority of whom are combustible, and many of whom get exhaled carbon monoxide. But if you look at the data on that as on its impact on behavior, it's actually not strong in the randomized clinical trials. It is reimbursable, though, so it can be offered for almost everyone. In this situation, are you recommending for our 4,000 users that we offer this test now, knowing that the great majority of those who are disadvantaged or at higher risk really couldn't afford the test, and so until reimbursement moves, which requires the evidence, what would you recommend?
Right. So at this point, we get into the whole ugly thing about reimbursement of who's going to pay for this. So at this point, this is not an FDA-approved test in that sense of there's a whole series of things that need to occur for it to move in that direction. 23andMe has been moving these things towards wellness reports, which are these kind of this other area. So this is the thing that keeps me up at night, because actually when we look at who is getting this genetic testing and who is finding out information about their different susceptibility, it's people who have resources. And we know that actually smokers tend to be in groups that have many fewer resources. So this is a systems issue that we need to address. I think that people deserve to know their genetic risks more and more. The price, again, has been dropping. As we get it into the medical field, though, the price will increase. So we have this whole conflict going on here of how do we do it? It is going to be a paradigm change, because how we've done things before has been you go to a doctor, you get your genetic test, and you have your one-on-one counseling. This is where 23andMe kind of blew us out of the system, where it's like you spit in a tube, you mail it in, you go online, and you get this information. And that's how we need to be thinking. How do we get this information out there easily with all this different type of consulting, all this different type of counseling? So I don't know. I think at this point, my simple answer is it's not reimbursable. It's not ready for prime time in the medical system. And it'll go out there faster just by direct-to-consumer, kind of like electronic cigarettes.
But the people who would benefit the most from it will need the most intensive behavioral pharmacotherapy.
And so many people are getting no pharmacotherapy, no behavioral therapy at all now. So yes.
Thank you.
A short question. David Sachs again. 30-odd years or so ago, the focus was on alpha-4 beta-2 nicotinic receptors, and I didn't hear that mentioned at all today. Do they have any role, or are they out the window?
Little itty-bitty role.
Itty-bitty, okay.
Itty-bitty role. Alpha-5, really big.
Got it. Thank you.
That's scientific talk right there.
Hi, June Feng from Centers for Disease Control and Prevention. I have a question on the genetic variation on the race, ethnicity, and gender, age, those demographics. Is it associated with those demographic information?
So I think the question was kind of, are there other characteristics that are associated with this variant? So we see nothing with gender. So male, female, we don't see anything.
That's what I expected.
Don't see anything with age. It's just, we all kind of age. Hate to say that. And, really our gene distribution hasn't changed. So, we see this change in smoking behavior over the last 100 years. Our genes haven't changed during that period of time. Huge racial differences. So this amino acid change, which is present in about 50% of white populations, is only about present in 5% of Asian populations, only present in about 3% of populations of African ancestry. The other variation of the expression is common in those populations. So still this gene cluster is contributing in the different racial groups, different world populations, but it'll be different combinations of variation in there. This keeps me up at night too. So because what keeps me up at night is that these genetic studies, 96% of genetic studies over all of medicine have occurred in populations of European ancestry. The major fear that I have is as we move forward with precision medicine, we are going to increase the disparity of care because we do not have the knowledge of populations of Asian and African ancestry. And the Asian, I'm not as concerned about the Asian ancestry because there's lots of consortia working on it with China and other regions, but the African population really will be left behind, and that has the potential, again, to increase the disparity of care in the United States.
Thank you.
Hi, Dr. Bierut. I'm on-body Global Medical Director at GSK Consumer Healthcare. We manufacture and market Nicorette and Nicoderm. I wanted to thank you. This is fantastic research, and congratulations to you and your team for what you found.
Thank you.
Wanted to know, what do you believe is the impact of your research on the ultimate goal of cessation?
Yeah, this is what my husband asks me every night, as a public health researcher. He's like, so what? That
well, that's not what I meant.
It's like, what do you care about this? So what I think, so I get to stand up here and talk philosophically. This is like my day. What I think is happening is when I think about what's really been driving smoking cessation in the United States, it has been the environmental changes that we've had. That we've reduced, the acceptability. I remember the times of sitting on an airplane, being in the hospital, people smoked, all these things. So what we've really done is we've really reduced smoking in the population because of, these environmental changes. We've had, pharmacologic issues, pharmacologic treatments. We've also had, our counseling. But in the wild, most people are smoking, going through smoking cessation on their own without anything. So what I want us to do is I want us to move forward with this science of saying that not everyone is equally vulnerable to heaviness of smoking. I think that this is something really important for the FDA, for the tobacco companies to know, in the sense of, how do we regulate for our vulnerable population? I think it also it's going to be a paradigm for how we use genetic information about behavior in general. What I hope, again, is that the numbers of smokers continue to decrease because of all the work that we are doing in our public health aspects. And, I think of smoking as the 20th century major illness. Obesity is the 21st century major illness. So I think the other impact is going to be, how do we use this as a model, this genetic information, to help drive behavior change? So I think that at some point we are going to have some better pharmacogenetics too, of understanding, which medications people should start on first. But I think that in the end, that's going to be less of a driver.
Thank you.
One more question. Okay, I was.
Okay.
Go.
Ted Lipinski from Battelle in Columbus, Ohio. Very great presentation. Thank you so much for sharing it with everyone. My question is about the biochemistry downstream from the genes. How much is known, and like as an example to help you understand the question I want to ask, one possibility is that everyone with the genetic discrepancy metabolizes tobacco smoke constituents in a certain way. Another possibility is that there's a diversity in how different people, even with the same genetic discrepancy would metabolize the constituents. And if there is diversity, that opens up complexity in cessation treatments. Like, we've identified that you've got, like, broadly speaking, we've identified that you've got a problem. We could fix the problem with this, or this, or this, et cetera. So those are the details. Again, biochemistry, how much is known?
So the variation in the nicotinic receptors is really not changing the biochemistry. It's really changing the function of the receptors. So how much is conducted, how much of the receptors are there. So it's not changing what I would say is the overall biochemistry. Nicotine metabolism, on the other hand, the other experts are here. I saw Rachel Tyndale, Neil Benowitz is here, and I'd rather let them answer that. So that my understanding is there are some different paths. So I think that's the short answer. What I also want to say, it's a general comment, which is I don't want to think of any of this as mutations. And I always use the word variation here, because we're sitting around, if we don't smoke, we have this variation, and I think we're all kind of normal, I hope. And so it's this diversity that we have. And I want us to think of this variation in that sense. So,
thank you.
Thank you so much. I really appreciate this.
Laura said it was her day, it was our day to be treated to that presentation and dialogue afterwards. It was wonderful. Break is outside. Please go to the exhibits. And just one more round of applause with bells, please, for all of our awardees this morning.